In question 9, we're told that insulin is a protein involved in the regulation of human blood glucose levels, and that genetic engineering allows us to create large-scale production of human insulin. We're then given 5 statements, A through E, and we're asked which of them describes the process of genetic engineering, in this case described in these 2 sentences. So we have our 5 statements, and as you can see, they're quite lengthy, and this sort of appears as a wall of text in the exam, and that can be quite disconcerting. But the main thing to do is to identify that they're actually quite similar statements, all of them, going through them, and there's only a few areas in which they differ. In fact, there's only 2 key variables at play here. And I'll talk through them as we read our first statement. So, taking insulin from a human and inserting it into the DNA of a bacterium, as the bacterium reproduces, it makes large quantities of insulin DNA that is used to treat human diabetes. Now, there should be a few things that we notice are wrong here. Insulin, as we're told, is a protein, and you can't insert a protein into DNA. You have to insert the DNA of the insulin, and then the synthesis can then take place after translation and transcription from the DNA. So, you can't take insulin and put it into DNA. So that's wrong. Moving on, as the bacterium reproduces, it makes large quantities of insulin DNA that's used to treat diabetes. Well, DNA doesn't treat diabetes, it's the actual insulin itself, the human insulin, which treats the diabetes. So we can say that on these 2 counts, this statement is wrong, and we can keep our eye out in the other statements for similar mistakes. Let's move on to statement B, which says that taking insulin from a human and inserting it into the DNA of a bacterium is what happens. Well, again, as we said, you can't insert insulin, a protein, into DNA of a bacterium. So that's wrong. So we know that B is wrong again. Let's have a look at the second sentence, though. As the bacterium reproduces, it makes large quantities of insulin, the protein itself, that's used to treat diabetes. That part's correct. So even though the statement as a whole is wrong because of the first sentence, we can look out for this second sentence being correct in the other statements. Moving on to C, is this the right one? Well, let's have a look. Taking the insulin gene from a human chromosome, that's much better than what we had before, and inserting it into the DNA of a bacterium. As the bacterium reproduces, it makes a large quantity of insulin DNA, so we've got that same mistake again about insulin DNA, which isn't used to treat diabetes, it's the insulin itself that we're looking for. So that's statement C being wrong. Moving on to D, taking the insulin gene from a human chromosome, okay, so that's correct. As the bacterium reproduces, it makes large quantities of insulin, so that's also correct. So we've got our statement that's correct, which is our answer D, and we got that by identifying the mistakes earlier on. Moving on to E, let's just double check here. Taking the insulin gene from a chromosome, well, that bit's correct, but then it makes the same insulin DNA mistake, so we know that E is wrong, and the only answer that's correct is going to be option D.